again, thank you very much for organizing this.
Thank you.
That's cool.
All right, thank you guys, and thanks for coping with as we ironed out some kinks and getting the webinar going. But today we have a wonderful webinar for you on presenting the future forward in flow cytometry, remembering the past, celebrating the present, and shaping the future. We have a great lineup of speakers who are renowned flow cytometry experts within their
All right, I just had a meeting come up, so I'm going to see if I can't get on. All right.
Robert, can you please place yourself on mute? Thank you. So before we begin, I want to review some quick webinar etiquette. Please mute your microphones. You may remain on video if you would like, but it's not required.
You going out?
No. Robert, please place yourself on mute. Please place any questions you may have in the chat. However, we will hold all questions until all speakers have presented at the very end. Each speaker will be presenting for about 20 minutes, and we do have 5 speakers presenting today. A panel will be held at the end of this presentation. All of the presentations will have a panel at the end, so you may raise your hand during the panel session, and I will call on your name so you can ask your question or share your comment. If you do need to leave early, please go ahead and place that question in the chat, and I will make sure that we do ask it while the panel session is recorded. So today's speakers, we have 5 speakers presenting. Our first speaker will be Rachel Walker. She will be talking about a brief history on flow cytometry. Our second presenter will be Rachel Sheridan. She'll take us on a journey through a flow cytometer. Third speaker will be David Leclerc. He will be talking about spectral flow as a transformative technology. Our fourth speaker will be Matthew Cochran, and he'll be talking about cell sorting from a 10,000-foot view. And finally, Tim Bushnell will talk us into the future is now. So our first speaker is Rachel Walker. She has a PhD in clinical engineering from the University of Liverpool, where she was introduced to flow cytometry. She's currently the director of the Byram Institute Flow Cytometry Core Facility. She's the chair of the Mid-Anglia Cytometry Club, an organizer of Flow Cytometry UK meetings, a member of ISAC and several task forces and committees. She was recently elected to the ISAC Council and is also a member of the Royal Microscopy Arts Society. And she was also awarded a BBSRC modular training program grant in 2014 to develop flow cytometry courses, to which she and Derek Davies have trained over 1,500 scientists in the last 8 years. I did put a link to her website for her flow cytometry core at the very bottom, if you guys like to take a snapshot at it and contact her in the future. So I'm going to turn the slides now over to Rachel, and she's going to talk to us about a brief history of flow cytometry.
Can you see that slide okay?
Yes, I do.
So good afternoon, I'm Rachel Walker. I'm the head of flow cytometry at Byram Institute in Cambridge. I've been doing cytometry for over 20 years. I started using a sorter during my PhD, and I found that learning the principles of how the sorters and analyze worked allowed me to design my experiments better, give me a greater understanding of the technology. So today I'm going to give you a whistle-stop tour of the past 60 years of flow cytometry. I would admit that there was a lot more than I could have put in this presentation, and I urge anybody who's interested in knowing more about the history to read Harry Shapiro's excellent Practical Flow Cytometry Bible, and also the Purdue Cytometry website has an excellent section on history, including videos from many of the founding fathers of flow cytometry. So what is flow cytometry? It's a technology that's been around for 60 years. It takes physical measurements on individual particles as they pass through a light source. It takes the measurements on every single event, and it looks at the scattering of light as well as the particle passes through the laser beam, and a measurement of emitted photons from the process of fluorescence. This can be fluorescence that is emitted from multiple fluorochromes excited by multiple laser beams. We have a Bigfoot sorter in our lab here at Byram with 9 lasers and 55 fluorescence detectors. So in theory, we can look at 55 fluorescent parameters off every individual cell that passes through the machine. When I say cell, because the majority of our applications are using cells, but flow cytometry can be used for beads, organelles, or in fact, any particle. It's a fluorescence-based technology. So the golden rule is if it's fluorescent, we can see it. As the particles pass through the light sources one by one, flow cytometry is the ultimate single-cell technology. Thousands of cells can be analysed in a matter of seconds, giving robust statistics very quickly. And there are thousands of flow cytometers worldwide in research labs, clinical labs, and biopharma labs. Many of you will use the term or know of the term FACS. This stands for Fluorescence Activated Cell Sorter, but actually it's more properly known as flow cytometry. FACS is a trade name of Becton Dickinson. So if you use the term FACS, it's a little bit like saying you're hoovering when you're using a Dyson. So the best way to describe your experiment are you're using flow cytometry, and they could be either analyzers or sorters. So where did flow cytometry all begin? I'd like to take you back to the beginning of flow cytometry and how it became a popular technique. I'm not going to talk about the fluidic dynamics work of Lord Rayleigh or the laminar flow work of Crossland Taylor, or even the work of Robert Hooke and his microscope, all of which contributed to our first flow cytometers. Instead, I'm going to start with Wallace Coulter. Wallace Coulter worked with his brother Joseph and founded the Coulter Corporation, which became Beckman Coulter in 1998. And he was the inventor of the Coulter counter. The urban legend that went around the flow world a few years ago told of Wallace working in the basement of his house, looking at designing an instrument to size particle pigments in paint, but found that his paint was frozen. So looking around for what he thought would have the same viscosity to paint, he thought he would try blood, and the Coulter counter was born. However, in reality, Coulter was aware from his pre-war visits to the Far East, the hours spent by medical technologists doing chamber counts of blood cells, often with poor accuracy. The atomic bombing in Japan and the aftermath made Coulter realise the need for rapid and accurate red cell concentrations, not just for individuals, but for populations, as an index of the recovery from radiation exposure. The Coulter principle and the Coulter counter device size and number of cells based on changes in electrical impedance when cells flow through an orifice. The cells are suspended on one side of the orifice and sucked through by a vacuum. And as they're sucked through, there's the electric current that passes through them, and this voltage is measured. As the cell passes through the orifice, it obstructs the flow of the electrical current. So we can measure the electrical signal that comes from that. If a signal will then tell you if a cell was present, and the amplitude of the signal will tell you the volume of the cell that went through. This machine allows you to obtain a distribution histogram that represents the size of the cell and the range of the population. And Coulter counters are still used in hospitals worldwide today to do complete blood counts. Also looking at the results of atmospheric nuclear testing fallout was Mack. He was a physicist working in the group looking at nuclear fallout in milk, meat, and other dairy products in Los Alamos in New Mexico in the US. One of Fulwiler's colleagues was looking at red blood cells on a Coulter counter and could see 2 populations and wondered if one of them was immature red blood cells. Fulwiler didn't believe that there were 2 populations, so he started to think of a way to separate out those populations. He wanted to pick out the abnormal populations to get the same distribution when looking back through the Coulter counter to prove his colleagues wrong. He started looking at valves with electrical switches to move a stream of flowing liquid from one channel to another. This was a very slow process. Then he saw Dick Sweet's inkjet printer paper. This paper from 1965 in Science showed an ink writing oscillograph. It applied Rayleigh's law that a stream of fluid wants to be hydrodynamically stable, so it will form droplets. Vibrating the jet of ink droplets in a regular pattern and charging those droplets meant they could be deflected onto a piece of paper. Fulwiler saw this as a way of moving small volumes of liquid quickly. So Fulwiler used a Coulter counter to look at the size of the objects and then charge the droplets containing the cell of interest into the tubes. His patent in 1965 covered the capture of analysis of the stream of flowing cells. Incidentally, he managed to prove his colleagues wrong. There was only one type of red blood cells present. So Len Herzenberg was an immunologist based in Stanford, working with his wife, Leonora. He saw the need to be able to look at immune cells in a high throughput manner, given quantitative data using fluorescence. Frustrated by his poor eyesight and the hours spent staring at cells using a fluorescent microscope, he started to look for other options. As an immunologist, he wanted to look at and sort live cells for immunological genetic experiments. Using a microscope, he could see different types of antibodies based on their ability to bind to different antigens, and he wanted to test this clonal selection theory, which meant that cells that were bound to a particular antigen should be able to make antibodies to that antigen, and cells bound to a different antigen would make antibodies to that antigen. The theory being that cells making the antibodies to the antigen A won't make antibodies to antigen B and vice versa. To test this theory, they felt it was necessary to separate out those cells, and those cells needed to be alive so they could be put back into a mouse or grown in culture to make an antibody A or B. The initial experiments were looking at binding the cells to plates using antibodies, but they couldn't get the cells off the plates in a viable form. So the Herzenberg started to look at options for cell sorters. Len approached Mack Fulwiler about his sorter, the sorter based on Coulter volume, and asked if fluorescence could be added. Los Alamos weren't interested in adding fluorescence, so they gave the plans of the machine to the Herzenbergs, and the engineers in Stanford built a sorter based on Fulwiler's machine and added detectors for fluorescence. Herzenbergs had seized on a visionary idea to build an instrument capable of identifying rapidly sorting live cells with distinctive surface molecules detected by fluorescently tagged antibodies. The combination of monoclonal antibodies conjugated to a fluorescence tag in the combination with a fluorescence activated cell sorter or FACS machine meant that multiple parameters could be looked at the same time. Now, the sorters of today in our labs are not much different from those early machines. We still use the principles of charging droplets containing cells and to deflect them into tubes or plates. The Herzenbergs worked with Becton Dickinson to commercialize the FACS machine, with the FACS-2 machine being released in 1976. It was launched with a one laser measuring forward scatter and fluorescence from the 530 nanometer laser, meaning that we could use fluorescein as a dye. The FACS-4 was released in 1979 with a second laser, the additional 568 nanometer krypton laser, meaning that the second fluorochrome rhodamine could be used in experiments. BD at that time had a business plan. They wanted to sell 50 cell sorters to the labs worldwide. Now, we know those sorters have evolved over the next 40 years. The principles are exactly the same. The machines have more lasers and more detectors. The first sorter built by Byram Institute was in 1982. We think it was a FACS 420. And this picture has been in my lab for many years, so I've always assumed that this has been our first cell sorter. And you can see that it's actually quite a large machine compared to what we're used to today. So flow cytometry has been instrumental in many applications over the years. They've helped to aid the design of the cytometer as well as making key scientific advances. So here are just a few. The Lawrence Livermore National Laboratories in the mid-1980s created the first high-speed cell sorter prototype. These were created to help the human chromosome separation in order to carry out research, especially the Human Genome Project. It was imperative that individual chromosomes were isolated before sequencing. These dual beam droplet sorters were capable of creating droplets at 220,000 droplets per second, sorting 20,000 events per second. The combination of Herc's 33248 and chromomycin A3 and high-powered lasers allowed individual chromosomes to be identified and separated, as shown in this figure. These machines led to the MoFlow high-speed sorter, which was launched in the late 90s, and then the influx sorter, and then to all stream and air systems that are on the market. These are important pieces of equipment for core facilities as they can sort faster than cuvette-based systems. The MoFlow sorters from Cytometry were also used for sex selection. Cytometry, in collaboration with Colorado State University Research Foundation, created XY Inc. in 1996. And using these high-speed cell sorters, it was possible to separate out the X chromosomes in sperm in cattle. In the dairy industry, farmers want female cows for milk production. With a 50% chance of female cattle being born, this has led to the culling of a lot of male calves. So by sorting out the chromosomes, it's possible to purify them with a 4% difference between the DNA between the X and the Y sperm. This is done using a combination of scatter and Herx 33342. This is a really big business now worldwide, not just for bovine, but also for other species. Before flow cytometry, autoradiography was used to look at cell cycle, and this was using radio isotypes. But flow cytometry offered cell analysis in far greater speeds. Herzenberg did a lot of pioneering work on the cell cycle using flow cytometry, using ethidium bromide and propidium iodide. And at that point, propidium iodide wasn't commercially available. Fixation was required. So Krishan used PI in a hypertonic citric solution that disrupted the membrane, so fixation and RNase treatment wasn't required. There was also an advancement when the UV-excited DAPI was introduced to flow cytometry. In the 1980s, with the onset of the AIDS pandemic, patients presented with dramatic declines in immune response and increased incidence of opportunist infections. Flow cytometry, still in its early days, was used to find the first clues of the causes of this disease. There was a spike in the number of scientific publications at that time using flow cytometry. And flow cytometry is still used today to detect HIV and for diagnosing when HIV becomes AIDS by looking at CD4 counts. This is still used as a clinical diagnosis tool, especially in sub-Saharan Africa. The AIDS pandemic has driven the need for low-cost CD4 tests, especially in Africa, where over 20 million people are infected with HIV. There is a need for cheap, precise, accurate CD4 counts. So let's hear from President Obama.
The work you do here is remarkable. And I just got a fascinating tour of your vaccine research center. I have to say I was very impressed with how you can clone a virus gene into a vaccine vector, then subject it to gel electrophoresis, and then pipette the samples into a 96-well microplate. Run it through the world's most advanced multi-parameter flow cytometer. I mean, it's impressive. I've been tinkering around the White House setting up a similar system. We use it for brewing beer, but it works well for your work also. The work you.
So President Obama wasn't wrong. Flow cytometry is used for brewing beer. In fact, many food industries use flow cytometry to look at yeast levels in beer and wine, also looking at levels of contaminating bacteria. It's used in the water industry to look at water quality. In fact, flow cytometry is used everywhere. Cytometers are placed on ships to look at levels of phytoplankton in the world's oceans. There's even a cytobuoy that collects and measures regular seawater samples. A microflow cytometer was placed in the International Space Station. Due to the anti-gravity conditions in the space station, the microflow was specially designed to work in a no gravity to reduce air bubbles in the fluids of the machine. And the astronauts used this system to study the immune responses while in space. Flow cytometry is used to look at the blood doping in athletics and cycling. It can only be used to look for unique antigens of red blood cells to prove if blood doping has taken place. The biggest drive in our technology has been trying to get as much information as we possibly can about every single cell that passes through our machine. So this figure was taken from a paper in 2012 showing the number of fluorochromes used in flow cytometry over the years. And it used to be that fluorochromes drove the need for new hardware. So as I mentioned, the first cytometers had one laser with one dye, fluorescein. Two lasers meant we then had rhodamine added. Then in the 1980s, we had PE and APC as antibodies, sorry, as fluorochromes to antibodies. And then the tandem dyes of the 90s really opened up the number of colors we could do from each laser. I remember Mario Roederer's paper from 2005 showing 17 colors. And this was quite a groundbreaking paper done on an LSR2. It used the tandem dyes and Q dots to be able to maximize out the number of detectors that we could use off that machine. And in 2012, it was claimed that 18 colors was the maximum that could be realistically done on a flow cytometer at that time. Then came the Cytop machine. This is a cytometry by time of flight. It combines flow cytometry and mass spectrometry. It was launched in 2012, designed by Scott Tanner and developed for immunology by Gary Nolan's lab. Instead of using fluorochromes attached to antibodies, it uses stable metal isotopes. The latest models use isotopes 79 to 209, which means that in theory, 130 parameters can be simultaneously measured. In reality, people use between 35 and 45 metals in each experiment. However, the Cytop is an inexpensive machine to set up and run. It requires a dedicated argon gas supply, a dedicated operator, and often a dedicated bioinformatician. The technology behind fluorochromes has increased massively since 2010. The Sheridan dyes were purchased by BD, who invested in the technology of polymer dyes. These are very, very bright dyes. They're a chain of polymers. They look like the angel of the north. They work like a lightning conductor and they pass their energy along the chains to fluoresce brightly. They took over from the quantum dots and the violet laser. Then the UV brilliant dyes were released that opened the UV up from the traditional 2 colors to 6 colors. Then there was a brilliant blue, brilliant yellow, green. Other dyes that have been released recently, such as the photonics dyes, which are now part of Thermo Fisher, these are the Nova Flow dyes, have introduced even more fluorochromes to the market, allowing more fluorochromes and more parameters to be used in every single experiment. So we look at the cytometers that were on the market. The fluorochromes availability drove the hardware developments. The fact Vantage of the late 90s was a 12-color sorter based on the PE, FITSI, APC dyes and their tandems. The 18-color LSR used those tandem dyes with the addition of Q dots and then the brilliant violets. The Symphony that was launched has up to between 5 and 7 lasers and can look up to 50 colors, utilizes the brilliant dyes. However, one of the drawbacks of these dyes is the fluorescence emission, which can lead to a lot of spillover and spread issues. And then came spectral cytometry, also known as full spectrum cytometry. Now, spectral imaging has been around for years. Paul Robinson's group in Purdue University patented it in 2004, and he presented his work in an ISAC meeting. It has been licensed by several companies, including Sony, who launched the first full spectral analyzer in 2012. It was the Cytec Aurora in 2017 that really was the game changer for full spectral cytometry, a 5-laser system capable of seeing above 50 fluorochromes. Spectral cytometry differs from conventional. With conventional flow cytometry, a dye such as FITSI is excited by a 488 laser, and we collect its emission with a narrow bandpass filter. basically collecting the maximum peak of emission. These bandpass filters are often narrowed to minimize spectral overlap with dyes. With a full spectrum, the FITSI would be excited by all lasers and the emission collected over all detectors. In the case of the Aurora, that's 64 detectors, allowing us to see weak signals, and also allowing us to use dyes not possible together in conventional flow, such as FITSI and GFP, as they would have unique signatures. So now full spectral cytometry has led the way for more fluorochromes. Fluorochromes with the unique signatures to distinguish them, and fluorochromes for less common laser lines, such as infrared or the 320 nanometer laser. The Aurora, with its 5 lasers, allows between 40 and 50 colors. The first 40 color OMIP, that's the optimized multicolor immunophenotyping panels, which are peer reviewed and in the Cytometry Part A journal, was published last year. And the Sony ID7000 has 7 lasers, and the Thermo Bigfoot has 9 lasers. So only time will tell how many parameters we'll be able to do in the future. So what is the future for flow cytometry? If only I had a crystal ball, but I'm certain that we'll be able to individually analyze more cells or particles with multiple parameters with a reduced cost. The basics of our hardware of our cytometers haven't changed much since their invention. They still use the flow cell. Sorting uses the same principles of electrostatic drop deflection, and there will probably be a drive towards miniaturization of microfluidics. But flow will be combined with other techniques such as sequencing. We can see from this tweet from Gary Nolan, who fostered the Cytop in his lab and drove the technology behind him, that he thinks that quantum barcoding will replace the Cytop and high-end flow cytometry. This uses cell-specific oligo barcoding on each cell. He had a paper in 2020 which showed 50 antibodies and over 70 targeted RNA regions that could be used in mouse and human species. And about spectral cytometry, when asked on the Purdue list, Paul Robinson replied that he thinks it's the best thing since sliced bread. So to finish, I will leave up my contact details and invite you all to email me if you require any more information.
Thank you, Rachel. That was a fun journey back to history and brought me back to when I was doing flow cytometry. It was fun to be alongside a lot of those developments in technology. Our next speaker is going to be Rachel Sheridan. Rachel joins us from Van Andel Institute in Grand Rapids, Michigan. She's worked in a flow course since 2013, is a current ISAC emerging leader. She's also a member of the ABRF Flow Cytometry Research Group and a member of the board of directors for GLIFCA, which is the Great Lakes International Imaging and Flow Cytometry Association. She has a special interest in flow cytometry education, process improvement, especially when intersectionality of flow cytometry and other technologies can be combined, such as in genomics and metabolomics. So Rachel, I'll turn it over to you so you can take us through a journey through flow cytometer.
All right, thanks, Nicole. Are you looking at the correct monitor?
You are on the correct monitor. We're good.
Yes. Thank you. Zoom, I think, is getting smart. I usually have to swap it, but it figured it out today. All right, thank you so much for the invite. And today we'll kind of take a quick tour through a flow cytometer and how they work. I know Rachel Walker touched on some of this too. So I think we'll have just a fun little jaunt. Maybe. There we go. So I like to think of flow cytometry as kind of the original high throughput single cell technique. People and scientists have been looking at single cells for hundreds of years. When microscopists have been doing this for ages, but it takes a lot more time and they can't see quite as many parameters. So the flow cytometry is really able to help unravel complex biology in a shorter period of time. A lot of times we think about this like maybe looking for a needle in a haystack in the case of like minimal residual disease or like so rare phenotypes, but you don't have to have rare phenotypes. Not only can you look for your needle in the haystack, you can also just look through the haystack and figure out how much of which kinds of grasses were in the field that they used to make the haystack. So kind of the overview of the process. In general, we have a fluorescently labeled single cell sample. This single cell aspect is critical for flow cytometry. If you don't have it in a single cell suspension, it's not going to fit and it's going to clog the instrument and we'll have all sorts of problems. And that, to be honest, that is really one of the most challenging things in the entire process, is getting a high quality sample. And now this sample you can label fluorescently if there's something you want to learn about. So you can use fluorescently labeled antibodies to look at what proteins are being expressed. We have functional dyes if you want to know about metabolic health of the cell, calcium status, things like that. There are dyes that bind DNA, as Rachel talked about. So measuring cell cycle is something that we do by flow cytometry, both in the past and current. This is an assay that we do fairly regularly. And there are other dyes. My cheeky little phrase is, if you can glow it, we can probably flow it. So there's a lot of flexibility. If you think you might need flow cytometry or want to try it, and you've got a fluorescent label, it's worth talking to us. So we take this labeled cell suspension and we run it through our magic laser box, the flow cytometer, and it reads out those fluorescent barcodes that we built up. So our panels, right? So it's not necessarily one barcode per cell, but one barcode per cell type or per phenotype. The flow cytometer spits out what we would call a list mode data file. It's an FCS file. It has some extra information in there that's useful. And then we can take that and we can visualize this in a number of different ways. A lot of people are probably familiar with seeing dot plots, right? So you label one axis, how blue is it? The other one, how green is it? And you can separate out the cells and look for populations. With the advent of higher dimensional flow cytometry methods such as Cytop and spectral cytometry, we are now seeing it's getting a lot harder to try to do this type of bivariate analysis. We need a ton of plots and it's hard to keep track of. So now we're seeing a lot more dimensionality reduction and automated data analysis in flow cytometry. Okay, so my part of this is really, so like, what's in the box? What makes a flow cytometer run? I would say there are three main parts to every flow cytometer. There's a fluidic system. This helps move the samples through the instrument. The cells aren't going to walk on their own, and it makes sure that they're focused in the right place for analysis. We have an optical system. We have to be able to excite those fluorescent molecules that you've tagged on your cells. So we need an excitation source, and then we need to be able to capture and process those photons that come from those fluorochromes. And then we have an electronic system. And this helps convert these electronic systems coming out of our photodetectors into more human recordable and human usable data. The electronic system is also super critical for controlling instrument timings and sensitivities, especially when you get into cell sorting and you have to make very rapid changes on a very short time scale to ensure that you sort the cells that you want. In general, the electronic system does a whole lot of work, but it is the most black box part of the system to me. It's the one where if that's the thing that breaks, I'm making a service call. The other ones, we can do some more troubleshooting. So let's start with the fluidic system. We're called flow cytometry. There's probably something going on there. So what we use this for is what's called hydrodynamic focusing. Okay, so we have a sheath fluid. Sheath fluid can be some sort of PBS with or without stabilizers and antimicrobials. It sometimes can be DI water. I've seen people run full medium, so some sort of driving liquid. And we use this driving liquid to take your bulk sample of cells, so like all these dwarves in a barrel just kind of hanging around in their tube, and then it pushes them into a single file line and runs them through the system. Okay, it is a laminar flow system, which is why we can use DI water as a sheath without exploding everything. Okay? And so then it's going to take these, it's going to move your cells to that interrogation point in a fairly orderly fashion if your system is clean and your sample is nice. So here's kind of what a flow cell looks like. This one, oh, actually, I think this came from an LSR that is finally getting retired back at Wisconsin. It was one of the instruments I learned on, so I have a fondness for it. Okay, so these are built with this conical shape. Your sample gets introduced from a tube that would be down here off screen, and it comes up. The sheath fluid is pressurized and running on the outside. So as your sample exits the sample line, the sheath fluid kind of pushes on it and it pushes back just a little, and they find this nice equilibrium. So you have a core stream in the middle that has your sample, and it's surrounded entirely by this sheath fluid. So I also kind of think of it as sending the cells kind of on a log ride at an amusement park, or if you've ever taken a stick and tossed it in a river, you know, it's going to align with the flow and then there it goes. Okay. And so the ratio of the center of the flow cell that's filled by sample to sheath depends on the pressures that you're running these samples at. And so the higher pressure you run, the more volume per second you move, but you do it by increasing the width of that core stream, which is something that we pay attention to because it can have an impact on data quality. So the fluidic system, main goal is to take your cells from the tube, run them through the interrogation point, and make sure that they're focused in the right place. The optical system, this is where we get to follow the photons from where we generate them to all the way where we see them. So flow cytometers, we need an excitation source. Anywhere from typically 1 to 5, though as Rachel mentioned, we are starting to get instruments capable of more like 7 to 9. I think that this is going to be an interesting region of growth because we're going to need more fluorochromes that run off of some of those lasers. Right? So we have those excitation sources. We have optical glass to help us isolate wavelengths of interest. If we get a whole rainbow of colors coming out of the cytometer, we may need to figure out, well, this photon what wavelength photon comes from what color and how do we separate them out? And then we need to be able to detect the photons. Right? We have multiple types of photodetectors that you will see in flow cytometers. You'll see photodiodes typically associated with something called forward scatter. Photodiodes are not particularly sensitive, but if you'll notice, this guy is over here. It's looking at direct laser light. It'll see that just fine. Now for the rest of them, you're more likely to see something like avalanche photodiodes or photomultiplier tubes. These have more internal amplification of signals, and so they're better for the smaller numbers of photons you would get through the system. There is a special type of cytometer. There's an imaging cytometer called the image stream, which actually does use a CCD and takes pictures of your cells in flow. It's very cool. Right. So we can do two types of measurements with a flow cytometer. First of all, you get some measurements for free, right? You don't have to label your cells. When a particle interacts with light, it scatters some. Flow cytometers are set up to look at two different types of scatter. So the first one, forward scatter, which I mentioned, this is when we use that photodiode. And so the cell passes through the laser beam, and as it does, some of that light gets reflected, refracted, bounced around quite a bit. So we put a photodiode directly where the laser would be coming through if there were no cell. To prevent it from being smashed with the laser when there's no cell there, we put an obscuration bar. So as the cell passes through the laser, some of that light is refracted or bent at an angle where it can hit the photodiode. This is often called size or relative size. Be very careful. This is highly dependent on the refractive index of the particle that you're measuring. If you really need accurate sizing, this is not the way to do it. Okay. So the more photons that get refracted to hit this photodiode, the stronger the forward scatter signal. They are often, but not always, larger particles. Sometimes you'll also hear this compared to being like a cell's shadow. The bigger it is, the more shadow it has. But it's loosely related to cell size. The other place that we put a detector is we look at something called side scatter. So at side scatter, we're looking for light that comes out at 90 degrees. And so the more complexity of a cell, the more things inside of it that photons can bounce off of, the more side scatter a particle has. Okay. So you'll often hear this called granularity or complexity. It can also be impacted by surface features and many things. So these are all kind of relative, but there is some utility to looking at these morphological parameters. And it really highlighted when you look at something like blood. Here are some different types of blood cells. We have like lymphocytes. They're very small. They're mostly nucleus. The nucleus is round. They're kind of like little ball bearings, right? And then we have things like granulocytes that are just chock full of particles in their cytoplasm. and everything in between, different nuclear shapes, different sizes. And we can see those sorts of changes reflected in how these particles scatter their light. So down here, low forward scatter, low side scatter, not a whole lot of internal complexity, not particularly large cells. That tends to be lymphocytes. Monocytes, you're going to start to see a bilobed nucleus. So there's a little more inside for those photons to bounce off of. So they shift up a little in the side scatter. And granulocytes are the disco balls of flow cytometry. Their side scatter is incredibly high, often off the scale. Okay. So I find that if you're working with something like blood or bone marrow where you have such drastically different morphologies, this gives you a nice handle and a sanity check when you're looking at your markers if you're doing immunophenotyping. Say you were looking for T cell markers, if you looked at side scatter versus a T cell marker, you hope your T cells would pop out down here as pretty low side scatter. Right? So it's not 100% perfect, but it's a really nice tool to have. When you're working with cell lines, these do tend to be much more homogeneous populations. So the other type of measurement we make is fluorescence. Fluorescence measurements, we're going to be measuring whatever you put it on that cell. So we've got our excitation source, generally fluorochromes. They will absorb photon to go to an excited state. There's always some energy loss and wiggling, and then they emit a photon of a lower energy. It's nice in principle, but physics has decided to come knocking, and so there's more than one wavelength that can be absorbed, and there is more than one wavelength that these fluorochromes can emit. So what we end up with are these excitation spectra. So here in blue, this is a pretend molecule with a couple of excitation peaks and an entire emission spectrum. So we know we have these nice narrow excitation bands on flow cytometers because we're using lasers, but we still have to deal with the full emission spectra. And as the number of fluorochromes, the number of detectors has increased in flow cytometers, so has the problems that this causes. We have to deal with overlapping spectrum. So here's just a bunch of colors I threw on a spectra viewer, and you can see that a lot of them, there are photons that sit on top of each other. So if I had a flow cytometer and I saw a photon that was 460 nanometers, what color did that come from? Yeah, it could be kind of a problem. So we have some different approaches to take to deal with this. First of all, we can do some things in the hardware that make it a little bit easier to clean up. So the two approaches that we take in our hardware is one, we often not all cytometers, but most will spatially separate their lasers. So what this means is that if you have two colors with a similar emission profile but a different excitation peak, you can separate them by time. So if something emitted at 700 and one of them was excited by the violet and one was excited by the red, you would know when the cell was here and say, oh, that's my violet 700 emitter. And when it was here, you'd know that was my red 700 emitter, right? So we can kind of unstack them a little bit that way. But a lot of our colors also have the same excitations or similar excitation spectra. But if they have different emission spectra, now we can use specialized optical glass to tease apart different wavelengths and look very narrowly at different color bands. The two types of glass that you're likely to see in a flow cytometer are dichroic mirrors and bandpass filters. Dichroic mirrors, they take all the incoming photons, they have a nominal wavelength on them, and they split it in two, right? So they take that whole pack of photons, split it in two at that number, and some go through the mirror and some are reflected. Which ones go where depends on the type of mirror. So for example, this is a 500 LP or long pass. So longer photons go through it, shorter ones get bounced. Bandpass filters, instead of reflecting, they get rid of extra photons and they only let through the ones within their range. So you would read this 52550 is the center wavelength that it will let through is 525 nanometers, and the entire bandwidth is 50 nanometers. So you can look at optical designs or optical benches of flow cytometers and figure out what colors you're going to see where. And there's an optical array associated with each fluorescence collection point, or usually one per laser. So if you were to come into a lab like mine, I have these maps for all of my cytometers. right? So we split them out, we'll say like violet. Off the violet laser, we have these filters. Here are some common colors that run in each one. Regardless of the type of cytometer you're using, if it is fluorescence based, you probably are going to have a map like this. Even spectral ones will have a map. All 64 detectors on the Aurora, you can pick the one where you're likely to have a peak, and it helps with design. Now, where the conventional and spectral kind of differ a bit is how they distribute their filters and what they do with the data that comes off of each of them. So in conventional cytometry, they are typically designed to put bandpass filters near the peaks of very common fluorochromes, so the things you're most likely to use. Spectral cytometry is likely to more broadly distribute their filters. And then when it comes to the math on the back end and untangling our signals, conventional cytometry throws out a lot of data, whereas spectral cytometry keeps things, so even off-peak emissions. And you'll hear a bit more about this, I think, from David in a minute. So we can use these hardware these hardware approaches to untangle our overlapping spectra, but it's not perfect. And so if you're around a flow cytometrist long enough, I'm sure you will hear us start discussing controls, and especially the quality of the controls that are brought into the core. The two types of controls that you're going to see for multicolor flow cytometry are really single stain controls. These are controls that have been stained with only one color. Any more than that, it no longer works for this. And what it's going to do is it's going to provide our instruments and our software with the ground truth of what a given fluorochrome's spectrum looks like and how much it falls into each filter, both peak and off-peak filters. These really do need to be of high quality. If they need to be the exact same kind of fluorochrome that you use to stain your panel with, they need to be bright and clean. We have ways to do this. It's great. The other kind of control that you might hear us talk about when you get, especially in higher parameter ones, is the fluorescence -1. One of the effects of having so many colors and all of this overlap is that we now have additional measurement error. And what this causes is it causes some spread in our data so that it no longer, there's more, kind of ends up looking like background. And so these fluorescence -1 or full -1 controls are stained with everything except one color. So they tell us where the delineation is between positive and negative signal in a given channel. And they they show you this in the context of all of your other colors. So everything that's contributing error is there. Okay. So these end up being really important for flow cytometry because of how our systems are built and the physics of fluorescence. Okay. So next, electronic system. There's not a whole lot that I really want to go into this. We have them. They are very important. So we need to convert these photodetectors, convert what they're seeing into something that we can use. These days, most cytometers are digital. There are probably a few analog ones left out in the wild. They mostly use this route where they're going to amplify the signal. We're going to convert it. We have our analog to digital converter here. It's constantly measuring what's coming out of that PMT, and then we can process it, convert it to a log measurement if you want using a log lookup table, and send it to the computer in a way that we can see. Electronics are also where we control what we do and do not see. We can set things such as thresholds. So if you have a sample that has a lot of very small things like red blood cells and you don't want to see them in your data file, you can use the electronics to say, no, thank you, and then it just doesn't exist in the file or on your screen. And we can also take advantage of some of the characteristics of the signals that we get out of these to help us with the biology and to interpret what we're seeing. The one question that I get quite a bit is like, well, how do you know you're looking at just one cell? This comes up a lot, especially when we're doing sorting for single cell plate-based genomics methods. So I just kind of want to briefly mention it here if you see it. So we do something called doublet discrimination, right? If you have a single cell passing through a laser, as it enters the laser, you're going to start getting signal out of your photodetector. At some point, it's going to be as maximally illuminated, and then it will leave the laser and that peak comes back down. Now, if you have two cells stuck together, physically stuck together, because this is a flow system, they tend to orient more like a snowman than a sideways peanut. So you have this change. Again, the leading cell is going to enter the laser beam. We're going to start seeing signal. We're going to peak because our laser beams are typically not, like cells are typically bigger than our laser beams are tall. We get kind of this scanning system. So we max out our signal intensity for a bit. And then as the trailing cell leaves, it comes back down. So we can look at these two pulses look different, right? The height is pretty similar, regardless of whether we have one or two cells, but the width or the kind of the signal duration is greater when you have two cells stuck together. And so is the area of the pulse. Okay, and so we can use these, we can look at height versus area or height versus width or area versus width and look, and it can give us an idea of which cells are single and which ones are stuck together and allow us to get rid of some of those doublets and aggregates from our data. So the electronic system not only controls all of this, pulls our signals out, but we can also take advantage of exactly what it's measuring to get additional data and clean up what we look for. I haven't talked about the actual cells in a while though. So where did they go? Once we got them to the interrogation point, I kind of stopped talking about cells, didn't I? And where they went depends on what you're doing. If you're using an analytical cytometer, the cells are gone. They are probably sitting in a waste tank that may or may not already have bleach in it. If you're sorting, hopefully those cells that you wanted are in your tube or in your plate. Some will still end up in the waste tank. And the cells that you didn't want are probably now in that waste tank. If you're using different kinds of sorters, or if you're doing something like Cytop, exactly how the cells have been disappeared may vary, but whether or not they survive the process kind of depends on what you're doing. So hopefully that was a nice quick tour of what's happening inside a flow cytometer. And the really key points are that the flow cytometry is really upheld by the physics of fluidics, optics, and our electronic system. And that lets us look through so many cells so quickly and with a lot of parameters. Great.
Thank you so
Thanks.
much, Rachel. I think I'm now going to have the phrase, if you can glow it, we can flow it, stuck in my head. I love that. It's so
If you
catchy.
ever have to talk about like sorting and scatter, you can also throw in if it scatters, it matters, but I'm going to leave sorting to Matt in a few.
I love it. Our next presenter will go a little bit, we're south from Michigan, not too far. We're gonna have David Leclerc present. He joins us from the University of Chicago, where he's the director of cytometry and antibody technology. He has been the director there since 2016. He's also a member of the GLIFCA board of directors, and he leads the Chicago Users Group in Flow Cytometry, which has one of the most memorable acronyms called CHUG. His primary role is in acquisition of technology for flow cytometry and strategy development for adoption of new technologies within the University of Chicago's Flow Core. So David, I'm going to turn it over to you.
Well, thank you very much. Thank you everyone for joining us. My presentation is Spectral Flow as a transformative technology. I'm going to apologize for the use of a buzzword here, transformative, but I think it's in this particular case, it's important to use it because it allows it to synergize. The goal of this presentation is basically to convince you that while spectral flow allowed us to do very complicated experiments going forward, there's a bunch of tools that came following the development of this technology that makes the job way easier, way more manageable. And so we've seen this version of the graph presented by Rachel Walker a couple of times already. Essentially, we're looking at the development of the number of fluorophores linked with the development of the technology that we're using. And we could see that as we use more and more of the lasers, the number of fluorophores that became available for our users kept increasing. And around 2016, 2017, when spectral flow came about, at least in my mind, and I believe it's a common thought, that people were thinking, okay, we're probably reaching a plateau here. This idea of getting bigger and bigger machines simply not going to fly. That's when spectral flow came up, and it allowed a lot of development to occur. So that's what we're going to discuss in this particular presentation. I'm just going to step back. So Rachel discussed traditional flow stampers in details. I'm just going to give back the traditional paradigm, the main paradigm of this technology, where essentially within your panel you have a number of markers. Each of these markers will be allocated to a specific detector on the instrument. So in a sense, the number of detectors on the machine restrict what the users will be able to do going forward. There's a specific thing to keep in mind whenever we think about traditional instruments, is that whenever you buy the instrument, you make a number of decisions about what kind of panels are going to be run by the user. So for example, when I got the position in 2016, the first thing we did is buy a Fortessa X20 that looked pretty much like this. We had 18 fluorescent parameters to stick on the different laser beams on different detector arrays. And so this is the decision I made. We have on the blue laser 2 detectors picking up FITC and PSA 5.5, 3 detectors of my 561 laser, 3 more of the APC. The last 10 detectors were split apart between the violet and the UV line because there was more room on the visible spectrum, so we could use more fluorophores over there. The decisions were made by myself on the basis of what would provide my users the best tools to develop the best panels. For example, I did not include a detector to pick up PSI 5 because PSI 5 just never plays well with APC, which is present in pretty much any panel you can think of. There's no PSI 5.5 detector. That one doesn't play with PSI 5.5 on the blue laser. So you make these kinds of decisions that kind of restrict what your users are going to be able to run on these instruments. And so in a sense, you kind of bake in the type of panels that the users are going to be utilizing and restrict the type of fluorophores that they're going to be able to look at. Through the rest of this presentation, I'm going to talk about the blue laser, and here's why. This is a laser that has bugged me for many years. With traditional flow stampers, the advanced instruments, it's a laser that simply does not pull its weight, right? It doesn't do much. For historical reasons, it's going to pick up the forward scatter and side scatter, but we could put it on any other laser beam. In most cases, the only fluorophore we're going to be using of the blue laser will be their FITC and PSA 5.5. It used to do way more. The blue laser was used to look at P and P tandem dyes, but as soon as the 561 laser was introduced to the instrument, all that stuff was sent on a different detector array, and there was simply not much to use of the blue laser. So now we have a situation where my blue laser, which is fairly expensive, simply does not do much, looks at FITC and PSA 5.5, and there's really nothing else that to be used on it. It brings this weird catch-22 situations where I'm providing an instrument that allows the users to look at FITC and PSA 5.5. Therefore, my users are going to use these 2 fluorophore mostly, and there's not much push to develop different fluorophore that would emit in different other regions, because most of the instruments are basically look exactly like this. So there's no new stuff coming up. So this is pretty much what changed when spectral flow came about. We already explained the basics of spectral flow. So the main idea is that we're breaking this paradigm of 1 detector per marker. And now we're using the entire array of detectors in the instrument to measure the full behavior of my fluorophore on every single laser beam. And so you're going to make multiple measurements for each of the detector. When you put all these measurements together, you essentially get some kind of signature that will be specific to your fluorophore, and feed that to the analysis software that will be able to essentially parse the different photons in the proper bin using an algorithm called unmixing. It's very powerful. There's many great advantages of using spectral flow. The first one is that I never need to worry about my filter set ever again, or the number, how I allocate the detectors between my different laser beams. As soon as I can excite something, I can measure it. And so I don't need to worry about the way I design my instrument anymore. Secondly, because we can now identify different subtle signature differences between different fluorophores, we can still differentiate these two fluorophores and use them together in our panel. So, for example, APC and LX647 on a traditional instrument will be excited by the same laser, emits in roughly the same region, will go to the same detector, and so you can't use those two together. On the spectral flow, because we can identify these weird difference in the signature, that's going to be enough for the software to identify what photons come from which fluorophore and bin them properly. And so overall, and this, by the way, is not something we shoot for. When you develop your panel, there's so much overlap between those two markers that you do reduce your ability to resolve the dim markers and so on. But it becomes a tool that's available to you down the line as you increase the complexity of your panels. But overall, it allows us to manage large panels way more efficiently. Efficiently for two reasons. The first one, we can use pretty much the same fluorophores that are present in our fridge. As long as the signature is somewhat different, we're going to be able to use them. And so we don't need to wait for some magical new fluorophore to come out on the market to help us. We can get started automatically, directly after the purchase of the instrument. Secondly, it's worth remembering that the cost of the spectral flow Cytometer kind of break the trend that we were observing back in 2014, 15, 16. These traditional instruments, in order to look at more and more parameters, we're just piling on lasers and detectors on the instrument, and the price kept going up and up and up. And so to look at the biggest war machine I could imagine, if I remember properly, the range of the price of the instruments was roughly $1 million. Mass cytometry came about at the same time, competing with it, and the instrument was also several hundred thousands of dollars, like 700 or something. When Cytec came with the Aurora, suddenly we had a machine that was half the price, and so it became really attractive for multiple labs to get that instrument and start developing large panel and look at more complex biological phenomenons. So now we have a larger group of people buying into spectral flow. They are all like me, look at their blue laser and think this needs to pay off. And now on my blue laser, on the Aurora, for example, we have 14 detectors, so we have way more room to look at stuff. And so now we need more fluorophores to play with. The fluorophores actually came about fairly quickly. I'm not sure how that happened. I suspect that groups like BioLegend had a bunch of fluorophores that did not have a good viable pathway for financial success with a lot of their different markers. For example, if you have a really bright blue laser excited molecule that excites at 550, because I don't have the filter on traditional instruments, nobody would really buy it. But now suddenly with spectral flow, all these dyes that were passed on become relevant again and can be thrown on the market fairly rapidly. The table we have here is essentially the number of fluorophores, a really large amount of fluorophores available before, and a lot of fluorophores came out after the release of spectral flow. So by family, the organic dyes, we already had extra fluorodilyte and so on. Cytec released a bunch of CF dyes, and BioLegend increased their catalog of spark and fire dyes going forward afterwards. On the protein front, protein markers are still the explanation point of most panels. PAPC are still foundational, but not much came out to my knowledge. On the tandem front, there was a lot available, and there's still some more coming up with the new molecules that are developed, the new organic dyes, we see a trend now of these tandem dyes going to the far red region, allowing us more markers available in our panels. In terms of nanocrystals, Q dots came and went. They seem to have gone away, and now they seem to be making somewhat of a comeback. But Bio-Rad came out with their star brights, which not without knowing much of the technology, they're called P dots, and so they're now available. The claim is that they have a lower emission range, so less overlap between the different fluorophores. Organic polymers did great for us. So the Sheridan technology, the BV dyes, increase the number of markers that were available, made the violet laser a really strong tool in most flow stampers. So brilliant dyes, super bright, were available. Now we have Curavia dyes, which are tunable organic polymers, meaning you can have the same fluorophore but at different levels of intensity depending on your mark and on your panel. That may become something that's really interesting. And then there's obviously more organic polymers coming out all the time at different wavelengths available. DNA polymers is a brand new thing that came a few years back. NovaFluors are these cruciform DNA structures on which you can attach different fluorophores. They are also tunable, so you can add different numbers of dyes on those. And what else? Where there were already a bunch of markers that were present, but since we didn't have the proper detector set to look at them, they were simply not being used. So Alexa 4542 is a good example here, which is nice, small, dim fluorophore, but it's pretty useful in panels. You would never have thought of using it on a traditional instrument, but with spectral, it becomes kind of a nice a tool for your panels. And there's a bunch of new technologies that came out as well. And this is new technology in the sense that I don't know what it is. I asked BD what the BD real blue, real yellow molecules are, and they simply won't tell me. I thought we were friends, but that's okay. They don't. I would have told you, but here we are. So anyway, it's wrapped in corporate secret sauce, so we don't know exactly what they are at this point, but they are available to us. Put together, these new dyes are great. They allow us to exploit the unused part of the spectrum, and so we can look at more fluorophores and stack them on the arrays of different laser beams. They reduce the amount of spillover either by limiting the cross excitation between the laser beams. So this is the claim done by the NovaFluors, or narrowing the emission of the fluorophores, such as that's the claim for the star bright dyes. And then there's a bunch of other features that get improved over time, increased solubility, the brightness. For example, Miltony came with their Vio Bright, where essentially they managed to scotch more molecules of FTSE, for example, on your molecules, so they get brighter, they shine brighter when you run them on the instrument. There's a couple of caveats to keep in mind. A lot of these dyes require special buffers, additional buffers that you'll need to utilize to prevent non-specific binding. So Sheridan technology is known for that already. So you really need the special buffer to prevent those dyes to kind of attach to each other. The NovaFluor does the same thing. It seems like the DNA molecule is very sticky, will attach to presumably dead cells and so on. So those also require special buffer. The problem here is that as you increase the number of markers in your assays, you need to use these buffers, your staining volumes keeps going up and up and up. And at some points you're moving away from your optimal staining conditions. And so I think that's an area of the development where we should pay some attention. Obviously, with newer tools, the choice of markers is limited, so time is the best cure here. And we should keep in mind that there's some level of simple rebranding, the same molecule getting in the same name, or a substitute that have no real value or limited benefit as we move from one to the other. but overall, it's all very, very good. On that note of redundant fluorophores, obviously, there's going to be a lot of them that will look at pretty much the same. So, for example, we have 7 fluorophore here that pretty much peak in this exact same channel on every one of my detector array for each of the laser beams. However, keep in mind that even if they seem to have the same profile, some of them might have different properties, different brightness, different levels of spreading that will benefit your panel over other fluorophores. So some of them will have an impact. So it's kind of still nice to have this wide of choice to pick from. Hey, I just introduced Spectra viewers previously, which I think we should start using more than the traditional Spectra viewer that we had. You don't know how many emails I get from new users that ask, for example, is it okay to use something like FITC and BV510? If you look at that data on a Spectra viewer, the two emission kind of overlap. And so obviously the question is reasonable. But if you look at the information that you get from the Spectra analyzer, here we're basically giving you the information of the emission of each of the fluorophores, depending on the laser beam detector array. And so we can see that my BV510 emits mostly out of the violet laser and FITC out of the blue laser. And so it's totally fine to use them together. So those provide better understanding of the behavior of our fluorophores. Start using them more and more. Spectral flow also allows us to identify the bad habits that we developed over the years. I was telling you that the blue laser allows us to look at FITC and per-CP sapphire 0.5, and per-CP sapphire 0.5 is kind of annoying. The problem is that it has such a wide excitation range, it's going to get excited by pretty much every laser in your instrument, and will basically overlap with a number of other fluorophores that are excited by different laser beams. So we have the UV laser exciting my per-CP sapphire 0.5 at about 20% over here, which is a lot. And that will make my per-CP sapphire 0.5 overlap with BUV 737. Same goes with the violet laser and BV 711, P5 with the 561 laser, and APCLXS 700. So in my career life, if you will, I've seen per-CP disappear. I'm hoping we'll see the per-CP tandem dyes get spirited away over time. Okay, so now we have the ability to stack a bunch of fluorophores out of my blue laser. We have here situations where I managed to add 8 of these fluorophores, and there's still room. We have a gap over here, another one between per-CP Fluor 710 and BB 755. The question you might have at this point is, how is that going to look on my data? And before Spectral, there was no real way of kind of guessing how that's going to look like. With Spectral, there's a couple of index that came about that provides some kind of answer for this. So there's similarity index and the complexity index that we can look at. A similarity index is basically a score from 0 to 1. At 0, you're looking at 2 completely different molecules. At 1, you're basically looking at the same thing. And basically, it gives us an idea of a general idea of the overlap that you can expect between your molecules. When you have a pretty complex panels, you'll have a lot of these similarity index, and you can get to a complexity index that basically scales from 0 to presumably infinity. The general idea is the lower the value, the better your score, and the better your chance of resolving the different markers in your assay. One way to look at it, for example, Cytec recommends for a small panel on the 5-laser instrument, small panel would be 10 markers or so. The score should be around 2 to 3. With a large panel, 35, 40 markers, the score should be around 40 to 50. And so my panel of 8 markers stuck on the blue laser over here at 558, the conclusion is there's probably a better way of making that panel, although this is probably going to work anyway. At this point, my understanding is that these index kind of look at all these fluorophores as if they were all co-expressed on all of your cells. This is obviously probably not the case. You probably have some biological interactions between in your model that where some markers might be expressed on different cells. And so in those particular cases, even if you have a similarity index that's pretty high for 0.86, if these two markers are expressed on two different cells, they won't overlap and you're still good to go. So even if your similarity index score, the complexity score is pretty high, it's not the full story at this point. Which brings us to the last portion of my presentation, which is the online panel design services that are popping up pretty much everywhere. If you heard of Fluorofinder, that was the very first one that came about, and it allowed users to basically look at instruments, the way they were built, and pair the markers with specific fluorophores and see if it's going to work, and where to get these fluorophores from the different vendors. Nowadays, we see these type of tools popping up pretty much on all the manufacturers of antibodies or instruments. They seem to all have their own tool available. The one I want to discuss is the one that came with EasyPanel, which is a new group. I think it's I don't know. It's a student. Like it's a really, really nice tool, and it's a really interesting guy that developed it. They all work the same. I'm just going to highlight the main things that EasyPanel does that the other groups do not at this point. The first step whenever you design these panels is choose the instrument that you're going to be using. If you're part of a core, typically the core will have contacted the service, and you'll find a list of all the detectors, all the instruments available in that core on the website already, so you can pick exactly the exact instrument that you're going to be using. Second step is choosing the markers, and here we're basically selecting the different array of the markers that you're interested in your assay. This tends to be a pretty clicking heavy task. EasyPanel allows to import an Excel spreadsheet, so automatically populate all these markers. From there, you can select the expression level of each of these markers. EasyPanel used the information gathered by BD a few years back. They did a pretty great study measuring the level of expression of common markers in human samples. So if you're using one of these markers, it's going to automatically recognize it and automatically score that expression level. Otherwise, if it's a new marker or it doesn't recognize it, you simply can do it on your own. And from there, you can group your different markers by groups of co-expression. Third steps, typically on the other platforms, you'll have to now pair these markers with specific fluorophores. With EasyPanel, all we're asked is specific requirements that you might have regarding, say, your viability dyes, or if you want to exclude specific fluorophores. Percipes 5.5, Pack Orange apparently shows up a lot when you use EasyPanel, so you might not want to use these two because they're not very good. But then beyond that point, EasyPanel automatically pairs your antigen with number of fluorophores and does that by optimizing the score it's going to get from the complexity index and the total similarity score. And from under product here, we have a clickable link that will send you to the manufacturers of these different products that you have available. So, and from there, obviously, if you're not completely happy with the way things looks, you can go in and manually make tweaks to this panel and try to find better solutions going forward. So very easy way to get started with a pretty complex panel. Even if it's not the full panel that you'll use at the end, you'll have a lot of work done on your behalf from the get-go. This is the end of my presentation. The two ideas I wanted to bring up is Spectral Flow brought a bunch of new fluorophores that are great to use, are easy to use, and provide benefits with your panels. And along with that came the need for more tools that allows us to better prepare our different panels. I'm going to invite you to get more information on our website if you want to use this QR code. Otherwise, I will pass the torch to, I believe, Matt Cochran.
Yeah, we're going to pass the torch over to Matt. Thank you, David, so much. There's just definitely a lot of information and new things coming out of flow cytometry. So we're going to shift over toward the east a little bit into Rochester, New York, and have Matt Cochran join us to present on his topic of cell sorting with a 10,000-foot view. He's been involved with flow cytometry since 2003. He's currently the technical director for the University of Rochester Medical Center's flow cytometry resource, also a member of the GLFCA Board of Directors, the ISAC SRL Content Task Force, and a past member of the ABRF Flow Cytometry Research Group. So Matt, you can take it over.
Hi, everybody. Thanks for the introduction, Nicole, and thank you for having me here. I also want to, and to everybody attending, I also want to say thanks to the previous presenters who did all the legwork for getting this portion of my talk set up, because, you know, I can build off of what they said as I go through this. Now, I'm going to talk about cell sorting, but not specifically only, let me see if I can answer, there we go. Not only electrostatic cell sorting, but I do want to put this front and center. You saw these a little bit of this earlier, but the fact that it's been 60 years, this technology was invented 60 years ago is just amazing to me. And to think and how long we've how long this technology has been maturing, just really, really interesting. Before I come all the way back to electrostatic cell sorting, though, I want to kind of take a few steps back, basically. Cell sorting is not, in my opinion at least, not specifically about electrostatic cell sorting. There's a bunch of different ways we can approach this. Cell sorting is the physical separation of specific cells from a heterogeneous population or sample. There is single cell sorting, which is specific to electrostatic cell sorters. In this case, I am identifying a single cell based on some characteristics, sorting that cell into its separate collection vessel. It's actually not the most popular thing that we do. What we do more often than not is actually all forms of bulk sorting, in which we collect more than one cell into a vessel, such that we can do some downstream analysis with that. You know, it can be as simple as seeding a well in a plate with 10 cells to get them growing, or 100 cells, whatever it happens to be, or it could be collecting thousands or millions of cells in a tube for those downstream assays. So bulk cell sorting kind of is encompassing all of these technologies or these techniques that I'm going to talk about. Enrichment is bulk sorting, but it's kind of more focused oftentimes on greater yield and often again on lower purity. These are kind of categorizing the different ways we can approach this. These are not technical terms or anything like that. This is just how I think of them. If you think of them with a slightly different name on it, that's okay. With enrichment, you're often not identifying single cells. You're identifying some kind of larger characteristics, and you can often do a kind of sequential process where you do some enrichment with one tool and then move into the more single cell methods after that. Just kind of giving you an idea of the scope here and the planning that needs to go into effect when you consider these tools. Why are we doing this? We want to rapidly generate highly purified populations of cells that we can use for any number of downstream applications or purposes. Functional assays, of course, if you want to identify a specific cell type that you can see how its function plays out. Transplantation studies and tissue culture, as I mentioned earlier. Perhaps the most popular right now, at least the fastest growing, is the different sorting techniques for downstream genomic purposes. As the cost of these different genomic assays is coming down, the need for them, the desire for them is going up, and that's where the cell sorters come in. Cell sorting, I've said this in a number of different places, is cell sorting and flow cytometry in general are rarely kind of the end point of your assay. They're often a midpoint to get you to the end goal that you're really looking for. It allows us to be involved in a lot of projects, but not necessarily to be anybody's final purpose or final goal. We So different ways of doing this. So this is not an exhaustive list, of course, but there's things like density gradient centrifugation and elutriation, captured bead based assays. Acoustic field is something that I want to, acoustic field based assays or instruments is something I'm going to spend a little bit of time on. And then we're going to circle back into microfluidic chip and droplet based or electrostatic cell sorting. First, I want to quickly mention a couple of these techniques. Density gradient centrifugation. Everybody knows the term phi-call. It's actually that's a trademark term, but a liquid density gradient methods. Great for large volume blood processing and things like that, but not terribly pure. Populations coming out, they're a little bit finicky for people who've done this before. Serial centrifugation for small particle work. As far as I know, this is still the primary method for identifying or collecting small particles. When I say small, I mean in the 100 nanometer range. Some of the electrostatic sorting techniques can get there nowadays, but not nearly where we want to end up, I think. Centrifugal elutriation is a technique that I've personally never been a part of, although I've had some conversations. Peter Lopez was on the call, or was in the talk earlier. I hope he's still here. He turned me on to this, and one of these days I want to dive into this a little bit. But a method for pulling populations out via different centrifugation methods. These techniques are enrichment techniques. You're not doing single cell work here. These are enrichment only, but still very valuable in their own right. I'm guessing at this point, everybody is at least at some point done magnetic bead sorting. Really easy to use. There are very little equipment limitations, but a little bit. Great for large cell numbers, lots of different options. There's a picture here from Miltenyi Biotech, but there's a number of other options on the market. Positive selection methodologies is actual single target specificity. You are identifying a specific target on that cell and pulling it out based on that target, whereas negative selection is a purely enrichment kind of tool. That does not mean it's not useful, but it's not single target options. It's bulk collection either way. You're collecting everything of that type. Buoyancy beads, relatively new, last few years. The picture you see there is actually this vial right here. Kind of hard to see in the picture there, but I have this on my desk because it's a fidget toy plus a demonstration option for people, where these tiny glass bubbles basically are activated in some way and will link to your cells, and then your cells will float to the top. Your cells of interest will float to the top, and everything else will fall out or stay in suspension. Kind of an interesting way of approaching this kind of bulk capture. Acoustic field-based technologies. This is really emerging, and I'm just going to spend a second on it, but it's really kind of neat. Applying an acoustic field across the fluid while it's flowing through, you can start to pull populations out. It's really, I think, currently mostly about cell separation and kind of cleanup techniques. You can pull red cells away or pull dead cells away from your live cells. But I think the sky's the limit for this technology. I think it's really kind of really growing right now, and I look forward to spending more time with it and learning more about it. Especially pairing this type of tool with the different microfluidic options that are also really expanding. It's going to be pretty interesting to see where this goes. Next, we move into kind of the heart of this whole thing in terms from a flow cytometry standpoint, and that is first with microfluidic chip-based options that are on the market now. There's a few on the bottom of the slide there. There's others on the market. There's others out there, but this is just to give you a quick example. First thing I want to talk about is kind of where it's full chip or cartridge based cell sorting. In this instance, you are introducing a sample. Everything is done within the cartridge. You collect your population or populations potentially, kind of all within that. You're not generating any droplets or anything like that. It's all happening within the cartridge. Very few biosafety or contamination and carryover issues with this type of system, which is fantastic. Generally extremely gentle as well, which is also very nice. Usually single population, although I think that's changing at this point. They can be fast for enrichment though, so potentially you can do repeated sorts of the same sample to pull multiple populations out sequentially if need be, especially because of how gentle they are. Even if they're not super fast, because of how gentle they are, your cells are still in good condition. I think most of the options on the market now are relatively limited in terms of their fluorescence capabilities. Not all of them, but I think many of them are. So you are maybe a little bit limited in terms of your single cell detection, but these are single cell tools. You are identifying a single cell based on some characteristics that you've selected, probably usually fluorescence, and pulling those cells out. So these are true single cell options, but they are bulk single cell options for capture. Then we've got our droplet-based systems. In this case, again, we are actually generating a droplet of fluid, and in that droplet is our cell, or are our cells of interest. Broadly, there's two types of this of droplet cell sorters: the jet and air, in which the laser intercept happens in or outside the nozzle or in air, as you can see on the left-hand side down here. Our cuvette-based systems, where the laser intercept is happening within a cuvette prior to exiting the nozzle orifice. There are microfluidic droplet-based systems that are kind of basically like using a single-use flow cell. You insert the microfluidic chip cartridge, and the sort is performed, and then you pull out the chip and replace it with a new one for the next sort. Make sure you're keeping everything clean and stuff. Those microfluidic options can be electrostatic or not, but they are generating droplets. Again, the droplets contain cells of interest, and as the stream exits the nozzle, it is vibrating, as Rachel alluded to earlier, and our cells are kind of breaking off in those droplets, so we can sort them, charge them, and sort them downstream. Again, with the microfluidic options here, the biosafety considerations here are slightly variable depending on what tool you're using. I think that kind of goes without saying for most of these instruments, but in this case, if you are using an electrostatic microfluidic chip, you are generating potentially aerosolizable droplets, so you need to be careful in that instance. But they do have limited carryover and contamination issues because of the single-use chips. Couple of options here from Sony and NanoCell that we've identified that I've put at the bottom here. The Sony has multiple nozzle sizes, and which is nice to give you some flexibility. The NanoCell is a fixed nozzle size, and in this case, it's actually not an electrostatic system. You're identifying your cell within the chip, and it is coming out as a droplet and dropping straight into your tube. It's a single collection and dropping straight in. They're not charged or anything like that. So there's different ways of approaching the same thing here. Usually a fairly gentle sorting technique because of the lower pressure involved in most of these systems. You are still pressurizing and you are still breaking off droplets, but with the lower pressure, that's usually a little bit easier on the cells. You can do bulk or single cell in these type of situations, which is nice, gives you an added flexibility. And the fluorescence capabilities on these types of machines are expanding. So while you're doing single cell targeting, you are able to target very specific cell types or more rare cell types based on a number of markers rather than one or two. Electrostatic droplet base. So this is the kind of cell sorter that we speak of in our flow cytometry labs oftentimes, although they're all available and they're all potentially in our labs. In this case, the flexibility here, single cell capture or bulk capture. This is the only, the electrostatic and the microfluidic chip with droplets here are the only ones that you can do true single cell collection, sorting one cell into a well or one cell into some type of collection vessel. Very high fluorescence flexibility, tons of options on the market, which is fantastic. You could potentially sort as many as 6 populations at once, depending on the machine that you have at hand. Most are 2 to 4, and some of the newer ones are up to 6 populations at this point. High speed in terms of tens or hundreds of millions of cells per hour, and highly manipulatable and adjustable. This is where the real power comes in on these tools. We have so many options in terms of how we're collecting the cells, whether it be plates or slides or custom. We had a client where we had custom etched glass slides where we could sort into the etched well into the glass slide. Variable droplet size and collection volumes you can manipulate from these machines based on nozzle changes, pressure changes, frequency changes within the system. On the right-hand side, you'll see 3 different streams generated on one of our instruments based on changing the frequency of the vibration of the nozzle, same exact pressure and same exact nozzle, but changing the frequency of the vibration changes dramatically the size of the droplets and the speed at which you can sort. Now, this is not the easiest thing to do. I don't recommend everybody just dives into this, but you can set this up. Tons and tons of flexibility with this type of tool. Now, that's I think most of us would agree with that, but it comes with complications as well. As Stanley said, with great power comes great responsibility. Because you can change so many things, because you have so many options here, you have to make sure you get it all right. And it's not the easiest thing in the world. It's a relatively steep learning curve to operating a cell sorter effectively. It's not terribly difficult to operate a cell sorter in gross terms. What is difficult and where you really have room for growth is in getting into the fine details and understanding all the nuance and really making use of those capabilities. So lots of people are taught how to run cell sorters, and then really good operators can really push the limits with what they're capable of doing on these electrostatic cell sorters. There's a significant expectations versus reality kind of balance here. I think this goes for all of the tools that I've mentioned here, but I'm focused in on the electrostatic cell sorters at this point. Quite a bit of communication between the operator and investigator is crucial. In our hands, we here at the University of Rochester do exclusively operator-run cell sorts at this point. We don't very rarely train investigators to run their own cell sorts. So we do them ourselves. We do them in conjunction with the investigator, because of what I just mentioned. That the operation of these tools, it takes a long time to get really good at it. But that necessitates a fair amount of communication between us and the investigator. A lot of pre-sort requirements and post-sort requirements, as well as knowing what kind of downstream needs. Everybody who's done any cell sorting has seen a graph or an image kind of like this, where you're balancing recovery, speed, and purity in your system. I toyed with the idea of trying to mock up a T-SNE plot or something like that, a dimensionality reduction tool, because I think this is an oversimplification of the process here. There are so many inputs, there are so many variables that we balance when we do these sorts to try and get the most out of it for the investigator. But this is the kind of go-to option for most people. They see these the triangle here and balancing. There is no way to optimize everything simultaneously. There's a bit of a push-pull that you need to kind of go through. And each of the tools that I've mentioned is better or worse at some things, and you need to kind of balance that as well. Understand your capabilities and your instrument's capabilities in optimizing these things. Wanted to mention this because it's crucial, even though not always especially well understood. You cannot run, so in our system, we're generating droplets, right? We're generating droplets as they come out of the nozzle, and the cells are in those droplets. Now, if there's too many cells in there, you run the risk of kind of overloading some of the drops. The sample is not perfectly distributed within the solution such that you get one cell per droplet every single time. That's just not how this works. What you do end up with is small clusters and variable distribution, the Poisson distribution within your sample as you come out, which indicates that you need to kind of dilute things out such that you have one cell per every 4 drops, or even one cell per every 6 droplets that are coming out of your instrument. The more precise you need to be, the more pure you want your populations to be, the further you push this, even if you go one per 6 or 1 per 8 drops as you go further and further to get a more pure population. I mentioned pre-sort and post-sort information. There's just a lot to cover. We have long conversations with investigators when they come in, need to understand the experiment prep that they're undergoing, what types of controls they plan on bringing, the cell size that they expect, make sure we know what instrumentation we have available and then we can compare that with their needs, understanding what type of collection tubes they expect to use, what type of collection buffer they need to use. Sample prep, I'm going to mention again as we go forward here, is huge. Post-sort information, what type of downstream needs, but we still need to know this in advance of the sort. You have to have all of this lined up before you can start anything. What are your viability needs, or can you sort into lysis buffer? Can we change the nozzle size or to make a more gentle sort, or do you have specific needs for concentration for whatever reason coming out the other side? Can we do a post-sort with your samples? It's always nice at some point in the development of a project to do some post-sorts to identify that we're actually getting the populations of interest. A lot of things to discuss with the investigator, or if you are the operator and investigator, a lot of things you need to have lined up and organized before you sit down in front of the machine. I took this I wanted to pay homage to our friend Howard Shapiro, and also I came across Rachel Walker's blog post a little while ago about Howard's laws. The 0th law, that there is no magic. This was mentioned earlier today. There is no magic. These machines are not magic. We are working with physics and chemistry. There is a reason for why things are happening the way that they're happening. That goes along with the idea that these machines are not perfect either. You can't just you can't expect perfection. You need to understand the pluses and minuses. Shapiro's first law, that a 51 micron particle will clog a 50 micron orifice. This is about knowing your cells. On its surface, it's about picking the right nozzle for your experiment, but also it's very much about sample prep and your single cell suspensions that you're generating. And then the 7th law, that you can't make good data from bad data. Garbage in, garbage out is kind of the relation here. Sample prep is the single biggest factor that we have to understand. It's easier said than done, but if you can't get a good sample prep, a good single cell suspension, then everything else is going to suffer. And that's just has to be how it is. There's nothing we can do to get around that. So that's where the primary focus is oftentimes in troubleshooting. And then we move into instrument issues and how we can deal with that. That being said, while sample issues are a problem, cell sorting does affect the cells. You need to keep this in mind when you're doing your cell sort, and you want to make sure that you are thinking about that and when you're choosing your tools. In summary, there's a lot of options out there, so make sure you're really reviewing what's best for you. The tools are getting better every year, so please keep an eye on that. That's an exciting feature of what's going on. I didn't do this talk kind of as a buyer's guide or anything like that, but if you are in a position to be choosing your next cell sorter, keep in mind there are different types of machines for different outputs. If you don't need the latest and greatest, then potentially that's not what you should be buying. Even though the latest and greatest might be able to do some of those lower end things, that may not be your best option. So just keep that in mind and think about all these pieces as you are preparing. Okay, I'm going to cut it off there, and thanks.
Thank you, Matt. We are running a little bit over time, but I do want to add a small blip in saying that this session is being recorded. When we go to edit this session, we will cut it down into each presenter's section, so you don't necessarily have to filter through the entire length of the video. But that being said, we're going to keep ourselves in Rochester, New York, and we're going to have Tim Bushnell join us where he can talk about the future is now. Tim has a litany of experience in flow cytometry. He began doing flow cytometry as a postdoctoral student, was a flow director in 2003 at the University of Rochester, and then moved into becoming the director of the Shared Resource Technologies, where then he got ambitious and started to reorganize it into the Center for Advanced Research Technologies in 2021. He's had a large presence on ISAC and GLIFCA, continues to serve on those ISAC committees, and is also an organizing committee member of NERDS. Tim?
Thank you, Nicole, and thanks everybody for staying around for this talk, and well as the speakers who went before me. It's been a great time. Let me make sure my slides work because I'm not working. There we go. Come on. There we go. Ha. So what have we talked about today? We talked about the history of flow. We've talked about how a flow cytometer works. We've talked about the spectral revolution and fluorescence. We've talked about cell sorting. So what have we learned? One, cytometers are composed of three elements: of fluidics, and optics, and electronics. Two, flow cytometers work best on single cell suspensions. Three, we need some sort of label to identify our targets. We have to have some sort of detector to convert photons to electrons. And because of all this, we're able to isolate cells based on their physical and biochemical characteristics. So why do we want to push beyond the current technology? How do we combine this technology and the labels to push beyond the current technology? How can we overcome the single cell suspension requirement? Or as I like to say, what's next? So let's start with our interest in high dimensional flow cytometry. So we saw from Rachel's talk, Rachel Walker's talk, the history of flow cytometry and to where we are now, as potentially as many as 70 parameters with some of our tools. Having more colors, having more parameters allows us to look for more populations. We can look at a width, which means we can look at a lot of different populations. This is good for discovery work. This is good if you've got some samples you want to fully characterize the different populations. Or we can look at depth, which is to look at fewer populations, but look at them in greater detail to understand the biology of these cells. And as part of this, we can discover unidentified populations. Of course, I misspelled populations there, so I apologize for that. As Jonathan Irish calls it, the cyto incognito population. So what are our options? Well, we've talked about traditional flow. You heard about spectral flow. We've heard a bit about mass cytometry. Now, two other technologies, Raman cytometry is up and coming, as well as imaging cytometry. And if you know me, you know that I like toys, and I like tools, and I like to have a breadth of different options for our researchers to be able to use, because everyone has its strengths and weaknesses. And I'm going to emphasize what David said earlier. Spectral viewers are your friend. With more and more fluorochromes coming out, it's really important to look at what's available and where they're going to express where they're going to emit their photons, where they're going to be excited, because that can help you decide whether you can use traditional fluorescent flow or maybe a spectral cytometer. I'm not going to talk about traditional fluorescent flow. Rachel covered that really well. Spectral flow cytometry, the two big ones on the market, as David mentioned, was a Sony system. And the Sony system's cool because what they do is they actually take a prism and they bend the light to different areas of a detector. Here it is, a 32 channel PMT, where Cytec just put a whole bunch of APDs and lets you find all the, lets you capture all that light. Other systems like the Bigfoot has 50 plus parameter or detectors. And in fact, any flow cytometers you have can generate spectral data if you leave one or more detectors blank. So if you've got your 18 color Fortessa hanging around, if you leave a couple of channels open, you can still do some spectral cytometry. It's not as powerful as these other systems, and what systems will be coming down the pipeline will be interesting. Another tool that you heard briefly about was, what if we replace the fluorochrome with a metal tag, and we replace the detector, a PMT, with a mass spectrometer? And that's what mass cytometry is. And this is just an example. You've already seen this as well. And why do you want to use mass cytometry? Why would you consider it? Well, it's easier to design panels. There's a more predictable spillover. The stability of the samples is what's very impressive, which is We've had cases where people can send samples and they can last a week or longer after they've been labeled before they actually get read on the system. And this can include significant temperature changes. We had a sample once that got left on the tarmac over a weekend. FedEx was very apologetic. And when we got it and ran it, it was the controls were as good as previous runs. Mass cytometry allows you to barcode. This allows you to run as many as up to 20 or more samples in one tube at the same time to help minimize batch effects and reduce your antibody costs. And of course, there's tissue mass cytometry, which I'll talk about in a minute, because this helps overcome this idea about we need single cell suspensions. Of course, the cons are you need to invest in new reagents, you need to have a dedicated instrument and new infrastructure, you need to have a large user base, and your sample is destroyed. So we can't sort with mass cytometry. These other features here, you can overcome if you partner with laboratories that have this technology, because you don't necessarily need to have it on your site, because you can ship them off relatively easily. So do not be afraid of considering this technology if you don't have one on site. What's interesting, though, is this concept of Raman cytometry. So Raman spectrometry measures the inelastic scattering of photons. So this is not a case where the photon from your light excitation source interacts with the compound. It is just a natural product. It interacts with the Oort cloud, the electron cloud. So the molecule isn't excited, but there's some changes in the electronic cloud that can be measured. The beauty of this is it's label free. You do not need to add any fluorescence, any antibodies to be able to characterize the cells. While it is a relatively weak signal, there are a variety of techniques to enhance it. And it's also because of the ability, the technology is advancing to increase the speed and sensitivity. One of the things that Raman cytometry offers that the traditional flow doesn't is that we can measure metabolic compounds. We can measure lipids. We can measure total proteins. We can measure amino acids based upon the Raman spectra. So we can characterize cells precisely looking at new parameters. And in fact, this technology has actually been led to a cell sorter, and I'll show you that in just a minute. Now, you also heard about this. This is one of my favorite tools, the imaging cytometer. This is the Amnis image stream. They're now part of Luminex. This allows you to do up to 9 to 10 fluorescent parameters plus a bright field and a scatter channel. And of course, a picture is worth 1,000 words. So with this technology, it is high dimensional because each of these channels lets you can collect up to about 30 parameters, and then you can add more based upon how you analyze the data and how you generate masks and things of that nature. Very cool, very specific use. Each of these tools have their strengths and weaknesses, as I've said, and imaging cytometry, I think, is becoming new. It's having a resurgence, and I'll show you why I believe that's the case. First off is the Attune Cytopix, and this is by Thermo Fisher. They've added a bright field camera to the Attune. This system, and if you're familiar with it, uses acoustical focusing to let you run cells at very rapid rates, but the acoustic focusing keeps them in the center of the core stream. And this is just taken from the Thermo site because I don't have one in my lab yet. And you can see the bright field images of these different cells. This will facilitate your gating. This will facilitate characterizing doublets and true counting of your cells to make sure you've got all your populations. Coupled with the fluorescence that the Attune can generate. And it gets even better because BD has released the FACS-Discover S8 with their cell view. So this is a spectral cytometer, an imaging cytometer, and a sorting cytometer in one box. And this will let you take all of the tools that we've talked about and get a lot of valuable information, not just use whatever fluorochromes you want because we can do spectral, but we can take an image and we can see where that fluorescence might be located in the cell, on the cell, or even co-localized between two markers that will allow us to use that to identify and sort populations of interest. Relatively new, and I'm looking forward to seeing what comes out of this and trying to figure out how to get one for my own lab. So if you want to contribute, I'll set up a GoFundMe and we can see what we can do with that. But as I mentioned, the Raman imaging sorter has been developed. It's not been commercialized yet, but this is taken from this paper here. And they use the microfluidics that Matt had talked about, shown up here. And they're able to analyze the Raman spectra of cells relatively rapidly. And then they're able to characterize and identify what cells they want. And because of microfluidics, they can either put the cell of interest to collect or off to waste. And this is just showing you some data looking at 3T3 cells that have been treated over time. And you can see they're looking at just lipids and proteins in this case. And you can see how as treatment increases, more lipids appear. You start to see a decrease in the amount of protein, and you can characterize. It's not something that you can do with traditional flow cytometry, but very valuable nonetheless. And again, label free. So I think this is another revolution that we have to keep our eye on. But last but not least is this idea about overcoming the single cell suspension. We've talked about how you need a single cell suspension for good flow cytometry. Well, we also know that location is critical and that microenvironmental differences can have a dramatic impact on a cell's direction and fate. So how do we deal with this? We lose this when we go to solution-based cytometry. Traditional microscopy techniques are limited in the number of parameters that can be interrogated. I think I've seen a system, the best I've seen about 15, 16. That was a very specialized system. And this also leads to this concept of spatial proteomics. That is the idea of looking at the protein in a spatial context. You may have heard of spatial transcriptomics, and I'm a big fan of spatial transcriptomics, but at the end of the day, we have to remember that DNA makes RNA, RNA makes protein. Protein's the stuff that does the work. So we need to be able to look at the proteins to confirm whatever everybody finds in their spatial transcriptomics assays. And you can do that by mass cytometry. This is two different techniques that are competing techniques that are out there. This is the imaging mass cytometry technology that is commercialized by now Standard Biotools. And this is the ion path MIBI technology. Both of them give you the same output. The biggest difference between the technologies is how do they scan the image? How do they scan the cell? With the Standard Biotools technology is a very destructive process, whereas with ion path, it's a much more gentle process. You can go and rescan the cells at the end of the day. So we're right at the time, and so I think I just want to highlight this. Over 50 years, this as an old technology and mature technology, we continue to innovate. We we need to use our flow cytometry, our techniques to confirm data from things like RNA-seq experiments. Everybody enjoys doing RNA-seq, that's great, but at the end of the day, the RNA doesn't tell you what the protein is doing. Has the protein been modified? Is it phosphorylated? What is it doing? That's where our technology with flow cytometry becomes powerful. We can also obviously combine this with functional assays, be it things like reactive oxygen generation, calcium flux, et cetera. And of course, cell sorting and cell isolation is an entry-level platform to get into many downstream applications. And we start to, the single cell world, single cell genomics, single cell proteomics is really dependent upon what we can do. And I think at the end of the day, this idea of meta-omics, combining all these various different types of data to get a full, complete understanding is where the next big step is. And so I think that's going to require us to understand not just how our instruments work and optimize those, but understand how we analyze these complex data sets in ways that we can interpret and understand. So with that, I'm going to say thank you. I kept us on time. Nicole, I'll kick it back to you once I figure out where we go.
Thank you so much, Tim. This has been an absolute treat listening to all of you talk about flow cytometry from past to present. Made me reminisce on my days of when I worked in the flow lab. It's been an absolute blast. So I know we're at the end of time, but if you do have a moment to stay online and would like to ask any one of our speakers a question or provide a comment, or just, get their contact information so you can move them online, please feel free to do so. Go ahead and enter your question in chat or raise your hand and I can find you and call on your name. So we have One question from Thomas, and it says, what should I look for when buying a flow cytometer for a small startup lab? Something like a Curie C6 is a good call, or it's better to pay for a service like the Byram Institute. Thanks.
I'll
Anybody want to take that one? Rachel?
So my experience has been that a flow cytometer works best if it's used a lot. So it's a fluidic device. And so if it's left not being used for even a day, you can end up with PBS inside there crystallizing or bubbles forming. And so then when you turn it on, you tend to have however long you've not had it on for's worth of problems. So I remember using a sorter that was barely used, and every time I turned it on, there would be problems with the lasers, or there would be big air bubbles stuck, and it would take hours and hours to get them aligned. So to answer your question, if you're going to do a lot of flow cytometry, I would invest in your own cytometer, but do be aware that a cytometer is not just the investment in it initially. You will probably need a service contract every year, and that tends to be at least 10% of the purchase price. So it may be cheaper and actually easier to come and use a service like the ones we have at Byram Institute.
Especially when you factor in staff to run those instruments and keep them up and running. As Rachel said, when they're run, they're happy. When they're not run, they're not happy. And so having somebody who can also troubleshoot those issues is important. So, you know, we're core people. We think you should come to the core and not buy your own instrument. But if you are going to do it very, you know, a lot, then think about it. And there's a lot of tools out there, options now at that sub 100,000K price for instruments that compete with just the Accuray.
Yeah, I agree with that. And sometimes you want to slightly future-proof your equipment as well. So think about trying to get extra lasers and detectors, because it might be that you've got another assay next year, and it's probably cheaper and easier to get it in initially than it is to upgrade.
Absolutely.
And I probably speak for everybody in the panel, but if anybody has any questions about particular instruments or what they should buy, I would be happy for you to reach out to me. Just send me an email. I'd be happy to advise.
So we have another question from Alexander. Alexander is asking, is somebody sorting exosomes? I'm just going to default to Rachel Sheridan's comment. If you can glow it, we can flow it. So I'm sure somebody is doing something with exosomes, but it's a tricky.
It is tricky. So there's a lot published on analysis of them. There is a recorded, oh, Rachel, I don't remember which kind of talk it was at Cyto, but Vera and I were on a thing and we talked about what would go into sorting exosomes. So people are kind of doing it. It is very challenging. We are no longer in a situation where our laser is taller than our particles. So you have to deal with swarming. It slows things down quite a bit. So it is technically challenging, but people are starting to do it. So I won't say it's impossible, but I will say if you want to chat about it, we can chat about some of the aspects that you may need to consider that you weren't necessarily thinking about for sorting cell-sized things.
Any other comments on exosomes from the panel? Rachel answered it. All right. I see one general comment from Mike. Mike says, thanks for the great overview. Did my PhD at the One Laser Facts a long time ago, and he's learned a lot today as well. So I think many of us can share in that sentiment.
I came in with 2 lasers. I started with a caliber.
I was excited when we got the second laser on our caliber.
To be honest, I mostly did the LSR because I started with calcium flux and not phenotyping. I have to do things weird.
Yeah. Arthur has a question. If you need to use 2 different specialized staining buffers, for example, Brilliant Violet or Nivafluor, can they both be used together, or is sequential staining better?
Typically, you'll use them together.
Together.
So David, do you use half of each buffer when you do that together, or what? Because I haven't done that.
We've done it's basically blocking different things. So we go the full recommended volume, which is why, like I was talking about, that eventually, as you get to large panels, your staining volume is so high that it's annoying. So
Rachel has a future cytometrist sitting on her lap right now.
Oh, I was going to say, no, mine are in school.
The other Rachel, yeah, it might be getting close to supper time for her, so.
I did let one of my little ones QC a Cytoflex once.
That's awesome.
Got to train them early.
Absolutely. I remember when I was learning photometry, somebody had the flashlight model of a homegrown flow cytometer, and that's how they taught us the concepts of light and fluidics and optics. It was a really interesting experience to learn it that way. Great. Well, I'm not seeing any other questions from our audience, so I'll take this time to thank our speakers. Thank you so much to all of you for presenting. It's been an absolute pleasure working with you over the past few months, getting this up and going, and even learning more than what I knew before. So I appreciate all of your time on this.
Thanks, everybody.
much.
Thanks, everyone.